have the last session of the night. We're going to do LigandMPNN, the last model for the sequence design of this bootcamp. So honestly, LigandMPNN is not that different from ProteinMPNN. The main idea is still the same. We have a protein backbone. We want to generate a sequence for that backbone. However, now we also have additional context. We have context that comes from a small molecule. It comes from a nucleic acid. It comes from something else that can bias the sequence design. Okay, a few results before we get into what's under the hood. Why do we need another ProteinMPNN? Well, the main reason is adding the extra context can really help improve the sequence design aspects. It can really help improve its interactions with other molecules. Take this example. We have a small molecule inside a protein interacting with it. If we look at the sequence recovery for Rosetta and ProteinMPNN, they don't Rosetta can, but ProteinMPNN definitely cannot take into context those other small molecule atoms. However, if we use those extra small molecule atoms, we can have a dramatic improvement in the native sequence recovery within 5 angstroms of small molecule atom. So we could really start to look at the properties of these small molecules, maybe find some sort of nice interaction, and really improve the sequence design. Similarly, we can do this with other molecules, including nucleotides. A very nice improvement, nice 20% or so over the other 2 models without context, and similarly, a dramatic improvement for metals. I think this is probably the most astonishing and probably the most compelling reason for why having this additional context is important. Okay, how does it actually add this context to the model? How does it learn from this? So similarly to ProteinMPNN, we have our residue graph. Each residue has all these backbone atoms. They look at all the other backbone atoms, so we calculate all these distances that form the edges between our 2 residue nodes. Now we're looking at our 32 nearest neighbors, so everybody has 32 nearest neighbors, residue neighbors. We also, if there's a small molecule, if there's a nucleotide or something like that, we represent the atoms. We represent the nearest 25 atoms for every residue. So for example, this residue, the nearest 25 atoms or so would be connected to that residue node, so it can have that information about where they are, what they are, etc. The ligand graph contains some extra features as well, including the atom type for each atom in that ligand graph. In the ligand graph, each atom represents a node, and they're also passing messages between each other. They also have information how they are relative to one another. We have information about the connectivity, whether it's a double bond, triple bond, etc. And I believe there's also like charge information, and maybe whether it's a chiral center or something like that. And then there's also protein ligand graphs. So like I said, every residue is connected to their 25 nearest ligand atoms. They form another edge there. So it's really kind of 2 graphs, well, 3 graphs. We have the protein graph, residues connected to other residues, the ligand graph, the ligand atoms connected to the other ligand atoms, and then we connect those 2 graphs by forming edges between residues and the atoms. Okay, questions about like kind of the input representation?
So why is it 25 and 32 close in protein residue? Like why 2?
I don't think there's like a specific reason. They kind of had to arbitrarily choose a number. However, I think if you think about it, I think it kind of makes sense. Like this is a nice kind of sphere around a residue. But I don't know if they had like a direct reason. You want a lot of residues, but you don't want too many. So there's kind of a constraint on memory and stuff like that too, and speed. But
they probably played around
yeah, there's probably a little bit of hyperparameter playing around with. Yeah.
So if you have protein ligand graph, isn't the ligand graph kind of redundant when it comes to the information? Shouldn't it be implied technically by the protein ligand graph?
It can be, certainly. But maybe by adding these extra nodes into this hypergraph or whatever we're building, we can have some extra expressivity, extra complexity to our model that may teach us something extra about it. I agree, it could probably learn it if we excluded the ligand graph, but maybe just, like we mentioned before, provides that extra flow of information that may, make it easier to learn. I don't know.
Well, in the protein sorry. In the protein ligand graph, there's no information about the connectivity of atoms in the ligand, right?
That's true. But like, if you still look at it, you can kind of look at the relative distance between it. So it can probably still glean kind of that geometric information from it, but
Is it actually is the information about the connectivity of the atoms in the ligand actually relevant to LigandMPNN?
well, I mean, think about it. Maybe there's like the electronic state of a hydrogen, a double bond or triple bond may impact how or what kind of residues can be interacting there.
Okay.
So for ligand,
Oops, sorry.
less than 25 atoms. So all the atoms should be considered by every residue.
So yeah, every residue is looking at their nearest 25 atoms. So even if it's like really far away.
No, so the really far away residue is also considered.
I believe so. Which, I don't know, seems a little inefficient, I believe. Yeah.
I think this was already said, but is there any information also embedded regarding the ligand functional groups?
Ligand functional groups.
For example, I'm basing this off of like classic Rosetta ligand docking. There's some generic atom potentials that utilize functional groups in order to score them. I know
Okay.
we're only scoring here, but I was just curious if that information was at all embedded.
I don't think explicitly. You can kind of have similar information by embedding the atom types and their connectivity, but I don't think there's any explicit like functional group representation that's embedded in here. Yeah.
Does LigandMPNN move the ligand in this, or does it keep it in like one state?
It's completely fixed, just like the backbone is. So you know where it's binding, you know what chemical types are part of that ligand, and you know where it's situated with respect to the protein itself. You're just designing the sequence.
What if you have a really flexible ligand? Like,
Multi-state. So you can maybe create a conformational ensemble of the ligand in the pocket, and then maybe favor multiple states. Yeah, that's my best guess. Okay, cool. So really, the rest of the model is quite similar. Once we have all these information, we have our coordinates, our atom types, and the coordinates for the backbone as well. We encode the backbone, we encode the ligand graph, and all the information is passed between all these different node types. The edges are updated as well, similarly to ProteinMPNN. And also, we have the same kind of sequence decoder kind of setup, where we have a random decoding order, so we can have really any residue decoded first, and can maybe provide extra context rather than the left or right. So we have our protein sequence. As you guys have started to see with the repo, there's also a sidechain packing network. This is not LigandMPNN, but this is just another model, but it looks the same. And the only difference is kind of what it's outputting. So here they're outputting something else. We'll get to that on a different slide. But any questions about like the sequence design part? Because that is pretty much the same. Yeah.
I have two separate questions. So like first but similar. First is like if you have, for example, a cofactor that is bound to the protein, like in case of a heme, right? So do you have to just explicitly assume this connection, or is it just that you write in that there is a bond here and you have to deal with this?
I believe you have to implicitly just assume that they're connected. They're going to be close enough that maybe the model can reason that they're connected, but I don't think there's like an explicit, like a explicit ligand bond between the sulfur atom or something like that. Yeah, that's what I think.
And the second one is like, does it consider also non-canonical amino acids or some modifications of amino acid glycosylation and so on?
That's a great question. I don't recall if they trained like that using like covalent modified residues, but I think it's totally possible. And the reason I say that is because they can do something similar in Rosetta fold all atom, right? They had they represented the non-canonicals as just a ligand, and then they had a specific interaction, a specific residue to residue edge that was formed between a covalent modification residue and a regular residue. So it could learn extra information about kind of like what is there. So I believe it's possible. I can't recall if they trained like that. But I do know that in some cases, they took parts of the protein. For example, you could take a sidechain and crop out the backbone around it, and then represent that sidechain as an extra ligand. So it kind of gives you extra information because that sidechain's interacting with other residues in the protein. Maybe you can kind of treat it as a ligand to kind of get more data from the PDB.
But I guess that is extremely costly computationally, because the ligand is, in this case, is more expensive than just another residue, right?
Yeah, but like there are small molecules too. Like if you had like a tryptophan, that's not that small. Yeah, it's just extra information, I guess. Okay, so we had done the sequence, pretty much the same thing. We decoded it randomly, and it looks kind of like ProteinMPNN in that sense. Only we now have taken into the context all the other atoms that are also in the system. Similarly, the loss function is quite similar. Categorical cross-entropy loss between the predicted sequence and the native sequence. If it's glutamic acid, we bump that probability up. We try to minimize everything else. Yeah, any questions on that? All right, the sidechain packing network. So what is sidechain packing? The idea is given a protein backbone and maybe the other context, the small molecules, the nucleic acids, et cetera, that are around that protein, and the design sequence, so you already have a sequence assigned to every residue in that protein. What are the sidechain conformations? So predict those. The way they have set this up is they've done an autoregressive chi decoding. So similarly to the proteins where they decode one residue at a time, here they decode one chi angle at a time. So they predict chi1 for every residue, and then chi2 for every residue, chi3 for every residue that have them, and then chi4. The way they do this is a little interesting. They predict a mixture of distributions. They predict a von Mises distribution, I believe that's how you pronounce it, I don't entirely know, which is essentially a Gaussian distribution that's wrapped. So because chi angles are periodic, so if you flip something over at 2 pi, you have the same exact shape. Because they are symmetric, we need a wrapped version of this distribution. So this is a wrapped version of the Gaussian distribution. So what they do is predict three of these distributions, which includes a mean and a standard deviation, like a Gaussian, except for they're wrapped. And they also predicts the mixing coefficients. So how much of one Gaussian do we want, how much of the other? So it kind of can predict these different spikes in the actual distribution that you're sampling from. And then they draw a sample. Yeah?
Why is the wrap, why is the Gaussian wrap?
Yeah, it's because of the periodicity of angles. So if you think of like a sine, it goes up and then down, it repeats every 2 pi. Same thing with cosine. So it repeats up and down, and now you're essentially the same place you started. So there's that periodicity in these angles, which is why you need a wrapped version. Because if you go past 2 pi, you've gone back to the other side, right? Yeah.
I'm just confused. I'm reading they're calculating the four chi angles.
Yeah, some residues have 4 chi angles. Sorry? Some residues have 4 chi angles, some have 3, 2, and 1.
Oh, I guess 4 is the max.
4 is the max. Not every residue has 4.
Right.
Right. Yeah. So if it has up to 4, they predict up to 4.
I see. Yeah.
Okay. Here they're just kind of comparing some results of the sidechain packing network. They're comparing what happens when you. So here they're comparing just the sidechain packs within small molecule context. Pretty accurate. The fraction of chi1s and chi2s within 10 degrees for each of these models. Here they're just kind of predicting a sequence with ProteinMPNN and then a sequence of LigandMPNN. Sorry, my bad. This is the ProteinMPNN model. This is the LigandMPNN model. So the ProteinMPNN model doesn't take into account ligand, small molecule, or other atoms, right? It only includes the protein atom. So it's the ProteinMPNN architecture. Does that make sense? Okay. And then LigandMPNN does take into context those. So taking into context does improve performance, not as dramatic as sequence. So I think that kind of makes sense. Like the model can still implicitly reason if there's a nice pocket there where the sidechains kind of need to be. I think that's not terribly unreasonable assumption. And here they're just kind of comparing it to Rosetta. LigandMPNN's better at predicting chi angles than Rosetta. Similarly for nucleotides as well as metals, yeah, the performance improvement's not dramatic, but pretty minor, and it does improve things with extra context. Some limitations. The only thing I could really come up with is that it probably recognizes transmembrane proteins as well. So you may need a soluble version of it, but also, yeah, I don't know how of a limitation that is if you combine it with ProteinMPNN, because it has a soluble version of ProteinMPNN. So may not be a super limitation, but it's there. Okay, that's pretty much all I have for LigandMPNN. Are there any final questions?
Could you elaborate more what you mean by combine it with ProteinMPNN?
Yeah, so for example, you can take maybe an active site, redesign the residues of that active site, and then redesign the rest of the protein given the active site. When you're dealing with the transmembrane, you probably don't want to model all the lipids in the transmembrane. You can probably just tell ProteinMPNN to design the transmembrane helices or whatever. And then if there's a small molecule in the extracellular region or the intracellular, you can model that with protein or LigandMPNN.
I'm struggling to, and this is because I'm probably ignorant in this topic, but what are some practical applications and uses for LigandMPNN? Because I'm having trouble finding many, particularly in a biomedical space.
I mean, if you're designing a transcription factor that's interacting with DNA, you might need to take into account those phosphate backbones of the nucleic acids. A small molecule drug that's binding a receptor, inhibitor, something like that.
I think enzymes, right?
Enzymes. Yeah, that may not be necessarily a therapeutic, but yeah, another definite example.